So today's video is going to focus on the kidney. In relation to the kidney, there are four key topics that you need to be aware of. So the first of those is the growth structure. The second is ultrafiltration. The third is selective reabsorption. And the fourth is osmoregulation. So today's video is going to focus in then very briefly on the growth structure, just to remind you of that structure, because much of that is similar to what you learn at GCSE. So you may wish to get your notes back out and review these before this lesson. So to start off with then, in terms of the growth structure, we need to think about the roles with regards to homeostasis, and that relates to osmoregulation, so water balance, and then with regards to excretion and the removal of that nitrogenous waste, urea from the blood, and there is a link here to the liver, which we have just looked at. Now, in relation to this, we can then sort of look further into the functions. And when we look at the functions, we can see that there is a function of the kidney with regards to filtering. There is a function with regards to iron balance, and there is also a function with regards to water balance. So from a filtering point of view, the kidney actually filters 180 litres of blood per day. So that is 92-litre Coca-Cola bottles. So those really big bottles that you can buy. Although someone told me recently that actually you can't buy those that large anymore. But if you can remember back to the big ones, the big Coca-Cola bottles, 2 litres, 90 of those, or a bathtub filled right the way up to the overflow is the amount of liquid that is going through the kidneys every single day. Now, in relation to that, they only actually produce 1 to 2 litres of urine. So they have to bring back and reabsorb a very large percentage of the liquid that is being sort of filtered through them, the filtrate, every single day. So the way in which they're able to do this is because of the functional unit of the kidney, the functional unit being the nephron, and there can be up to 1 million nephrons per kidney, and that does vary from person to person. So as you can see here on the diagram, the 3 key areas of the kidney are the cortex, medulla, and then the pelvis. And you can see the nephron as the functional unit here overlaid. The nephron sort of sits with the Bowman's capsule in the cortex, and proximal convoluted tubules also in the cortex. Your loop of Henle drops down into the medulla, comes back up the medulla into the cortex, and then finally your collecting duct drops back down all the way through the medulla and into the pelvis, draining into the ureter in order for that to filtrate to then leave via the bladder. Then, as you can see detailed on the screen now, the functional unit of the kidney, the nephron, has a number of key sort of parts to it that you need to learn as well. For the purpose of today, we're going to be taking an overview of the nephron, ready to look at ultrafiltration in the next session. So if we just highlight in on the box, this is the area we'll look at predominantly when we look at ultrafiltration, and you can see here you've got the afferent arteriole bringing blood into the glomerulus, which translates as ball of yarn. That is encapsulated by the Bowman's capsule, and blood then leaves via the efferent arteriole, and you can see these capillaries then sort of network and weave themselves around the entire nephron. The nephron itself, from the Bowman's capsule, then goes into the proximal convoluted tubule, which, as we said before, both of those structures sit within the cortex. Proximal convoluted tubule then dips down the descending loop of Henle before coming back up the ascending loop of Henle, and this is in the medulla of your kidney. We then have the distal convoluted tubule, convoluted because of these loops that you see. So you can see it's meandering backwards and forwards, and those are why this is described as being convoluted. And this is going to be sitting back in the cortex of your kidney. And then finally, your nephron is going to feed into a collecting duct, And you can see here that you've actually got a number of nephrons that are branching off of that one collecting duct. And that collecting duct is then going to take that filtrate. Now, any remaining water at this point can be reabsorbed if there is antidiuretic hormone present to sort of pores into this collecting duct and allow that water to leave via osmosis into the adjacent capillaries. Otherwise, it's going to go then down and for the first time enter the pelvis. And at this point, that filtrate is now becoming urine, which is going to leave the body. So as it enters the bladder and then out via the urethra. So before the next video, I would recommend that you have a look at Module 3 and look back at the structure of the capillaries, as this is going to be really important when we start to look at ultrafiltration.